Welcome back to BioFarm 2020. Our next speaker is also in Ireland. It's Dr. Fiona Brennan, and her topic this evening will be managing soil biological health on your farm. Fiona is a research scientist with Teagasc, which of course is Ireland's Agriculture and Food Development Authority, and she leads the Soil Microbiome Sub-Programme. Fiona completed her PhD in microbiology in NUI Galway. And the key focus of her research team is on microbial soil-plant interactions in agronomic systems and on understanding how these relate to the function of microbial communities in soil biogeochemical cycles and greenhouse gas emissions. Her group also investigate the fate and transport of microbial enteric pathogens in the environment, antimicrobial resistance, soil biological health, and microbial adaptation to environmental niches. As Fiona will explain, agriculture is currently facing a number of very significant challenges. Principal among these is the necessity to develop climate-resilient, low-carbon agricultural systems of farming that are both economically and environmentally sustainable. I'll hand you over to Fiona.
Hello everyone, and greetings from Wexford. I'm delighted to be able to join you this evening. My name is Fiona Brennan. I'm a soil microbiologist working for Teagasc, the Irish Agriculture and Food Development Authority. I work in the Environment, Soils and Land Use Department in Johnson Castle. And what I was asked to talk about was some of our ongoing work trying to understand microbial communities in soil. Over the past few years, we've been looking at microbial communities in soil, the length and breadth of the country, trying to understand what organisms are there, what they're doing, and how they're influenced by the environment, the soil, and also how that soil is managed. And what we want to be able to do is distill the knowledge on these communities into practical sustainable soil advice. So for those of you not familiar, Johnson Castle, our research focus is on soils, nutrient efficiency, carbon sequestration, biodiversity, water quality, and gaseous emissions. The group I work with is focused on soil and environmental microbiology. We're one of the most recent additions to the soil programme in Teagasc. We're in existence about 4 years, but we've already established a reasonably big group. We have 2 permanent members of staff, myself and Aoife Duff, who's a technologist. And working with us is a really fantastic group of young researchers, all looking at different aspects of soil microbiology. This includes understanding the organisms on and in plants and within soil, understanding how microbial communities cycle nutrients, the role of microbial communities in generating greenhouse gas emissions, microbial functioning in soil, indicators of soil health and how we benchmark our soils. And we also work on human pathogens and antimicrobial resistance in soils. So our research interest really boils down to 3 main questions. The first of which is, how does agricultural management impact microbial communities? And that's the whole array of management that we can impose on a system, from fertilization to liming to the crops we grow, to cultivation techniques, to crop rotation, etc. All of these have the potential to impact soil microbial communities. And so what we're trying to understand is how exactly they're impacted and what impact that has on the overall functioning of soil. We're also interested in determining what constitutes a healthy soil microbiome. And again, how do we benchmark soil so we know whether the soil biology is healthy in one soil or not? Finally, can we harness microbial communities to make agriculture more sustainable? Can we develop systems that have we're using microbes to, I suppose, have healthier plants, to reduce the amount of inputs you have to put into the system, and also reduce environmental losses? We're not alone in asking these questions. In fact, there's a huge research activity going on looking at these questions internationally now. And the reason for that is what we're trying to, I suppose, contribute to is those global challenges that are facing our food production systems. These include, of course, the growing and sustained demand for agricultural produce, but also generating those produce in a way that protects the environment, that protects our soils, our air quality, our water quality, that maintains biodiversity. We need to reduce greenhouse gases and ammonia emissions. We need to control pests and diseases without chemical interventions, which are currently being withdrawn from the market. And all of this has to be done in the context of a rapidly changing climate, more extreme weather events, and issues around economic viability of some farms. In terms of the status of the environment then, on the environmental sustainability side, we're not doing very well just at the moment. Globally, we're seeing catastrophic losses in biodiversity. And indeed in Ireland, 36% of our designated habitats are at bad conservation status, and 15% of our designated species. Our rivers are decreasing in water quality, and we're losing those pristine water sites. Agriculture has been identified as the main pressure and threat. Then when we go to look at greenhouse gases, obviously being an agricultural nation, agriculture makes up a bigger proportion of our greenhouse gases than other countries, about 32% of our overall greenhouse gases and 98% of our ammonia emissions. But in the last few years, the trends in terms of emissions have been going in the wrong direction. While we did see a decrease at the end of the 90s, since about 2011, there's been a growing increase in greenhouse gas emissions associated with intensification in some sectors. But this is obviously a huge issue and a huge challenge for agriculture going forward to reduce those emissions. And to underpin the importance of this, I mean, last year, Ireland was one of the first countries to declare a climate and biodiversity emergency in our parliament. Then when we talk about soils and soil health, internationally, soils are under a lot of pressure, and we're losing huge tracts of fertile soil every year. Recent reports from the EU suggest 60 to 70% of European soils are not considered healthy due to current management, and a whole range of threats to soil health have been identified. In terms of soil biodiversity, we don't yet have a very good handle on what the status of global soil biodiversity is. There isn't sufficient data, but initial attempts to map where soil biodiversity may be under threat indicates that this is the case in a huge part of the planet. There's quite a considerable amount of our soils that are under pressure. And so all of this together, I suppose you come to the conclusion that our systems are not currently sustainable, and we need to change how we do things to better marry environmental sustainability and how we produce food. Change is coming, and internationally there's been a huge drive towards policies that will underpin green societies. In EU, we've seen a plethora of documents, strategy documents, roadmaps. We've had the Green Deal, we've the Farm to Fork, we've had the EU biodiversity strategy, we've a new roadmap for soils health with the soils mission. And they're all pointing in the one direction of travel. All and interwoven in all of the documents is the aims towards carbon neutral societies, societies that are not damaging to the environment, that reverse biodiversity loss, and that protect our soils. For those of us that are interested in soils, it's quite notable, I think, that one of the 5 missions of the next EU framework is on soils and soil health. And this speaks, I think, to the enormous importance of soil, not just in agriculture, but in broader society, and how we're absolutely dependent on them for a whole range of essential ecosystem functions. This includes providing most of our food, our fiber, fuel. They're also storing and filtering our waters, cycling nutrients, sequestering carbon, and acting as a home for biodiversity. Incredibly important functions. And when we think about whether a soil is healthy or not, we can think of it in terms of the capacity of a soil to deliver those essential functions. And for the most part, every soil has the capacity to deliver a multiple of those functions, but not to the same, each individual function, not to the same degree in each soil. So what I mean by that is you could have a soil that's very productive in terms of dry matter yield, but that same soil, its inherent capacity to filter water or to sequester carbon might be lower than another soil. So when we think about whether a soil is healthy or not, we need to take this into account. And one size does not fit all. What is healthy for one soil might not be healthy for another. Also, we need to take into account that all of these functions are important. And when we manage our soils, we need to take this into account. So soil health is made up by three components: chemical, physical, and biological properties of soil. And these are completely interrelated and influence each other strongly. On the biological side, when we think about soil health, soil harbors an absolutely staggering diversity and abundance of soil organisms, each occupying their own niche in soil. So this diversity incorporates a whole range of organism types, sizes, shapes, colors, all operating at different scales, from the microscopic, your small microorganisms like your bacteria, your fungi, your archaea, right up into our largest soil organisms, macrofauna, such as earthworms. These organisms, as I said, all are occupying different niches, and the diversity within soil is very much reflective of the dynamic and diverse nature of the soil environment. And this is important when we later think about how we manage those soils. Soil has a very pronounced spatial structure, meaning you get gradients of resources in terms of nutrients, of organic matter, of water. And this supports a large number of niche for organisms. As conditions change then within the soils, so too does the abundance, the diversity, and functioning of those organisms in soil. Of the three cogs of the wheel, if you like, the biological, the chemical, and the physical, the biological is the least well understood. We've always known it's been important, but the study of soil biology has been limited to a large degree by technical limitations. And I'm particularly thinking here of microbial communities in soils, which are the most diverse and abundant group of organisms. In the past, we might have tried to extract organisms out of soil and grown them on a plate in the lab, or put a suspension under the microscope. But in both cases, we only get to look at a tiny fraction of the overall community using these methods, making it very inadequate to look at the overall health of that community. But these technological limitations have been largely overcome by new methods coming on stream. And now, instead of trying to pull organisms out of soil and bring them into the lab and looking at small components of them, we can look at the overall community together in situ by extracting DNA, RNA, or protein from the soil, and using techniques such as qPCR or next generation sequencing. We can get genetic information from those directly. Then when we use bioinformatics, statistical or machine learning approaches, we can make sense of this complex biological information to come up with something sensible, which tells us about the composition of these microbial communities. And then taking a step further, we can look at these communities and how they change over gradients of environment or soil type or management. So which brings us to our research, what we're doing. We have a huge amount of projects undergoing at the moment, and we'd be here all evening if we were to talk about all of them. So I just picked out a couple to talk to you about in a bit more detail. So one aspect that we've been looking at is the impact of above ground diversity. That's the impact of the crop you're growing above ground on the below ground diversity, we'd have expected to have a very pronounced effect. So we've been looking at this with respect to mixed species swards. And there's been quite a lot of interest in these swards because they appear to have quite a few benefits over monocultures, in that you can put less fertilizer, etc., into them. They tend to be higher productivity if you get the mixes right. They reduce nutrient losses. They demonstrate higher resilience to climate stress and weed suppression. And although there's still questions to be asked in terms of persistence and how we best manage these systems, in terms of the low input systems that we need to drive towards if we're going to meet our environmental markers, these systems show a lot of potential. And I know there's a lot of farmers trying them out at the moment and successfully planting them. So what we've been interested in is looking at whether the above ground diversity affects the below ground diversity, and also is the microbial communities in those mixed species swards more resistant and resilient to climatic stresses such as drought. And one of our PhD students, Kerry Ryan, has been looking at this in 6 species mixtures. So she's looked at 2 grasses, Lowlium perenne and Timothy. She's looked at the 2 clovers, red and white clover, and she's also looked at herbs, which are chicory and plantain. And she's been looking at the microbial communities in a trial that was established by my colleague John Finn in Johnstown Castle, where we're looking at these individual species on their own in monoculture, and then mixing them in various combinations right up to and including 6 species mixtures. We've also incorporated paired plots so that one of the pair gets a drought for 9 weeks of the summer, and the other is rain-fed. And so what Kerry's been doing is looking at the effect on soil physical and chemical properties. And then she's looked at employing molecular methods. She's extracted DNA, and using sequencing, she's been able to look at the community composition of the microbial communities. Using quantitative PCR, we were able to look at the abundance of different groups of organisms for bacteria and fungi, for example. And we've also been able to look at genes, counting genes that we know are important in functions. So Kerry has been looking at genes that are important in the nitrification and denitrification process, and seeing how those are impacted by the above ground diversity. And finally, in addition to genetic potential, Kerry has also been looking at the capacity of those communities to deliver certain important functions using assays. So, for example, we've been looking at the capacity of the community to decompose, to respire, to mineralize nitrogen, to nitrify and denitrify. So before I talk about the microbial results, I just very quickly have a mention about the agronomic results. So this is the work of Guylaine Grange, PhD student of John Finn, on these very same plots. And what this plot shows is how the dry matter yield varies depending on the plant composition. So at the bottom here, we have our legend. And you can see that the colours represent the amount of tonnes per hectare dry matter. And the greener you go, the more dry matter you have. And these triangular plots up here indicate the plant composition and how the dry matter varies with the plant composition. So you can read this a bit like you would read a soil texture triangle, if any of you are familiar with those. At the edges, we have our 3 species types, functional groups. One is herbs, one is grasses, and one is legumes. And at the point, they are at 100% of the botanic composition. So here we have 100% legumes. And anything in between, anything in here, is a mixture. it's composed of different proportions of the different functional groups. And the real take-home message here, and what I'd like you to see, is that the highest yield is not coming from the monocultures at the point of the triangle. The highest yield is actually coming from mixtures of these proportions. And this is even more pronounced when we look at drought. So these are for 2 years of average 2 years of results. So these plots would have had 2 droughts over 2 successive summers. And here the sweet point in terms of yield is even more distinguished, if you like. And it's this point here in the greenest color. And what's very notable for me is that even with 2 droughts, this mixture here is yielding higher than a monoculture without any drought employed. So in terms of overall yields, these systems seem to be functioning very well. So what does the microbial results look like? So this plot here is very similar to the last one you've seen, except instead of looking at dry matter yields, what we're looking at is the number of bacteria in the swards. And similarly, we have our clover, we have our grasses, and we have our herbs. And what you can see is we're getting different numbers of bacteria depending on what the composition is. So the highest bacteria seems to be in the swards that are predominated by grasses. And when we move beyond abundance of bacteria, fungi, etc., we can look to specific genes we know are important in how the system functions. This gene here is called NERK. It's important in the pathway for denitrification. And again, we can see how the abundance of this gene changes with the composition. And it's a similar story to what we saw in terms of bacteria. The largest populations of this gene are occurring where your sward has predominantly grass in it. And then further, if we look beyond genetic potential to do processes or the number of organisms, we can look at the actual function of the community through assays. And this one here is an example which is looking at N mineralization. So we're looking at transformation of nitrogen from organic forms into plant available forms. And you can see that the capacity of the community to mineralize differs depending on what the plant community is. And in this case, it appears that N mineralization potential is higher, either where you have a lot of grass in your sward or very little grass in your sward. So we're still working through these results and we'll hopefully have this data set completion the next few months. But the preliminary results indicate that we're seeing strong plant identity effects, meaning that there seems to be specific microbial communities associated with different plants. And from a management perspective, then that means that if you have a field where you have all of these species growing together, you're probably going to get a much more diverse microbial community. So in terms of Kerry's question, does the diversity of the above ground plant community have an effect on the below ground community? The results so far indicate that yes, that is the case. So another project that I briefly mention is looking at the impact of fertilizer type and inhibitors on the soil biology. We mentioned previously reducing greenhouse gases is one of the biggest challenges facing agriculture. And we've been looking at a wide range of mitigation options for greenhouse gases. Research indicates that switching fertilizer type from CAN to urea, for example, can substantially reduce the amount of greenhouse gases being produced. And if you add an inhibitor, urease inhibitor, for example, to slow the flow of nitrogen through the system, those reductions in greenhouse gases can be even greater. But what we wanted to look at is what is the effect of these different fertilizer types and these inhibitors on soil biology. And we were fortunate that a colleague of mine, Patrick Forrestal, already had a trial running for a period of 5 years. So looking at a more long-term effect of these different treatments. The treatments include a control treatment where there was no fertilizer applied, a CAN treatment, a urea treatment, And then urea with the urease inhibitor, urea with a nitrification inhibitor, and a urea with urease and nitrification inhibitor. And my colleague Aoife Duff was leading this work from the microbial side. And what we're interested in from the microbial side was very similar to what Kerry was interested in with the mixed species sward. Is there a difference in composition, and is there a difference in function with the microbial communities associated with the treatments? So this data again is preliminary, but what I can point out to you initially is in terms of the abundance of our bacteria, our fungi, archaea, it looks like there's not much of a difference across the treatments. So this plot A here has the number, apologies, this thing, go back there. So this plot here shows the abundance of the bacteria. The red dots represent the bacteria across, and you can see they're all at pretty much the same level, so there's not a huge amount of difference in the numbers of bacteria across the different treatments. The green represents the fungal communities, and again, there's not much difference across the treatments, and the same for the crane archaea. So in overall terms, in abundance of different organisms, the treatments don't seem to be having an effect. When we dig a little bit deeper then and we look at the genes that are important in nitrification and denitrification, there's some variation, but in general, there's no significant differences across any of the nitrification or denitrification genes. So moving on then to the composition of the community, when we sequence that community and see who's there, these results are almost fresh off the sequencer, so we haven't got a chance to look in very big detail to them. But these plots represent the abundance. So each bar represents a single treatment, and each section in the bar represents a component of the community. And just by eye, you can see that the patterns across all of the treatments are very similar. We're seeing the same sort of abundance of the different groups. We will be looking at this in more detail and looking at the impact on overall diversity, but the initial indications are the composition doesn't change a huge amount. And then when we look at actual function, when we look at mineralization potential or the potential of the community to respire when we add a carbon source, when we take into account the variation between plots, again, there's not much difference. So to sum up, in terms of our preliminary conclusions, there seems to be similar composition and abundance across the treatments. Functional nitrogen communities are similar. We're seeing similar functioning across all the treatments. There is some impact on the end cycling when we put in the inhibitors, but that's what we would expect because the inhibitors are there to change the flow of the nitrogen. So briefly going on to another project that we have running at the moment. This is the PhD project of Aisling Moffat, and she's looking at biocontrol of leather jackets. So for those of you who are unfamiliar, the leather jacket is the larvae of the crane fly, also known as the daddy long legs. It's a major pest of grasslands and cereals in Ireland. Any of the chemical control options that were there have now been withdrawn from the market. So there's very few viable control options currently for this pest. Aisling's project is looking at trying to understand the occurrence of these pests. Where do they occur, what soil types, what climates, what crops, and also is there ways we can control them through understanding where they occur. So her first point of call was to do a survey across Ireland, and she also did a survey across Ayrshire in Scotland, looking at both tillage and grassland sites. She had a range of sites across the country on different soil types, different pHs, different soil chemistry, etc. She actually, unfortunately, got interrupted in the survey because of the COVID restrictions, but we're hoping to pick this up next year. So hopefully she'll be hitting quite a few other sites across the country. In each site then, she took plots, or she took samples across the plots, about 25 cores across fields. She looked at the leather jacket occurrence, the numbers that were there, and also what species were occurring. She analysed the soil, both in terms of physicochemical properties, and also she took samples for microbial analysis so we could understand the microbial communities co-occurring. She looked at vegetation analysis, grass heights, vegetation, and also taking into account climatic and management factors. So when she looked at all the data, the data we have so far, most of the occurrences is in permanent grassland. Looking at the soil chemistry, it seems that on the initial data, pH, magnesium, and phosphorus may be important in terms of the occurrence of these pests. And you can see there the pH plot that at the lower pHs, we seem to have more abundance. Now, we will need more data points to make this relationship, but initial look-see indicates that those parameters might be important. So as we sample more sites, we'll get a more clearer view of what these relationships are. And actually, if there are farmers out there who might be interested in having leather jacket surveys done on their farms, to take leather jackets, but also to sample the microbial communities, Aisling is still looking for sites, so you can get in touch with us on that. And further, what Aisling is just about to do is to look at the microbial communities. So we're interested in seeing, given it's a soil-borne pest, are there certain microbial communities that might be suppressive to its occurrence? And while the surveys have been stopped in their tracks, Aisling has been honing in on some of these aspects that might be important. pH was indicated in the survey that it might be important. So she set up some pot trials looking at the impact of different pHs on the larvae survival. These cores were taken from long-term plots in Johnson Castle, and she's going to be looking at how those larvae survive over the different pHs, what the impact is on the plant, the shoot and root, and also what the difference is in the microbial communities. The composition of the sward may also be important in where these pests occur, both in terms of the larval survival and also the choice the female makes in where they lay their eggs. So Aisling has been looking at the impact of grassland species on leather jacket survival. She's looked at the same species that Kerry looked at in the mixed species swards. She's looking at grasses, legumes, and herbs, monocultures of each of those, and also a 6-species mix, and then adding larvae to the plots to see how well they survive and the impacts on each of the plants. So that data will hopefully be generated in the next few months. And finally, Aisling is also interested in seeing if there's aspects of the soil microbial community that might be possible to be used as biocontrol agents. And to do this, she's been doing what's known as soil baiting. And in this case, you get an insect called Galeria. It's the wax moth. It's very susceptible to pathogens that attack insects. And you can put this into different soils and see how it survives, and if it dies, what might be the possible causes for it. So she's put these insects into soils from both mixed species swards and monocultures and see what the impact is on mortality of them. The main results so far are the main cause of death seems to be nematodes. She has isolated, cultured, and identified nematodes. And her next step then is to challenge leather jacket larvae with these nematodes to see if any of them might be effective biocontrol agents. So those are the main trials I'm going to talk to you about today. But I'd just very briefly mention, just before I finish up, a couple of new experiments that are getting off the ground that might be of interest. The first one is a project called FASTIN. It's funded by the Department of Agriculture. We often get questions about biostimulants, humics, seaweed extracts, a whole range of biostimulants, and there's not an awful lot of scientific data on those. So as part of this project, we'll be looking at biostimulants to see the impact on soil and nutrient cycling. We also often get asked about what the impact of slurry spreading is on nematodes, and that aspect will also be covered in this project. We'll also look more in depth at mixed species swards and the impact on the microbial community and cycling of nutrients, and potassium and sulfur on soil organic mineralization. So those are some of the things we'll be working on over the next 4 years of this project. One other project that might be worth mentioning is a project called MASTER, funded by the EU. And this project focuses on linking microbiomes across the food chain. So we know that a soil microbiome strongly influences your plant microbiome. And then when that plant is consumed either by animals or humans, it can impact the microbiome of animals and humans. So this project is about linking up all of those microbiomes across the food chain. And the aspect we're looking at is the soil microbiome. And we've just set up quite a big trial in Johnstown Castle where we're looking at a whole range of microbial bioinoculants, bacterial and also mycorrhizal inoculants, and looking at the impact of management and sward type. So we just sowed this at the end of the summer. So we'll begin to start monitoring this over to spring and seeing how those different inoculants impact our swards. So that brings me to the end. Just really to summarize the key messages I've been trying to get across is that there's an awful lot of research activity ongoing. This is still a science in its infancy, but the knowledge is building very, very quickly, and there's a lot of energy and enthusiasm in this area to develop a new understanding into the systems. And the new tools that have come on stream are enabling us to understand these microbial communities in far deeper depth than we've ever had before. Ultimately, the health of our soils will be determined by how they're managed. And we're hoping that this information can, I suppose, inform that management to give the best advice so the farmers can make the best informed decisions about how they manage their soils. And with that, thank you very much for listening, and I'm happy to answer any questions you might have.
Very comprehensive indeed, and we've had lots of emails coming in, lots of questions, so I'm going to dive straight into those. The first is from Shane, and Shane asked the question, what species perform well in your research of multi-species swards, and what species didn't perform at all?
So are we talking about plant species, or are we talking about microbial species? I'm not 100% sure on the question.
Probably plants, Fiona. Let's work on the assumption we're talking about plants, yeah.
Well, like the previous speakers, I suppose mixing plants together does give an enhanced yield for all of them. So they occupy different niches, they have different root architecture, they're able to access different parts of the soil in terms of nutrient acquisition. So I think they all do pretty well. As you go through the year, some plants become more dominant than others. I know in the drought that we had, the clover did fantastically well, whereas the ryegrass really struggled. But then it depends on the time of the year. Clover comes in at certain times, the grasses are more dominant. But altogether, they work as a pretty good team to have vegetation throughout the year.
Okay, and on that we'll come back to, we have a drought question coming up for you in a few minutes, so you've partially covered on that, but I'll keep moving. There's a question here from Tommy. It's a slightly tricky one. He asks, are the Teagasc tillage team going to rethink their strategy for high synthetic fertilizer and pesticide use now that we're more aware of the soil being a living ecosystem? And Tommy says that he's never gone to a field crops walk where soil biology has been mentioned, and it seems to be just NPK and pH that are mentioned when it comes to soil. So I know you're taking one for the team here, but I guess you're well placed to maybe address Tommy's concerns.
Yeah, no problem. So I think the area in Johnstown, we've been focusing on soil for quite a long time. Soil biology is a relatively recent addition. As I mentioned, we're only microbial group's only going about 4 years or so. So I suppose there has been, we certainly would take it on board. There hasn't been a huge amount of information coming out on the soil health front for the last while, but I think this has changed in the last 2 years or so. We had a really big soil health fertilage open day last year. I think we had over 200 people down at that. We've been putting a huge amount of effort into knowledge transfer, both the researchers and the advisory service, in trying to get those soil health messages out. We've had webinars. We just had a video released for Science Week. So I think certainly in terms of soil health is high up the agenda, certainly in Johnstown and in Teagasc. I suppose the other question that was asked was about adding fertilizers, reduction of fertilizers.
Yeah.
I think as I mentioned in my talk, change is definitely coming, and whether you're a conventional farmer, an organic farmer, or regenerative or biological or whatever, I suppose, brand of farming you practice, we're probably going to see a shift all the way down more towards the lower input systems all over, because there's no other direction to travel. So I think that'll more and more be incorporated into the advice, and I expect that all of I know our advisors and all of our team are getting trained in how to change their advice based on the best scientific information.
Okay, absolutely. I'll move on. This is from Mark G. Mark asks, are there any zero chemical fertilizer trials of multi-species in Ireland?
Zero in. I don't know, to be honest. I know actually one of your earlier speakers showed trials of Thomas Maloney. Those trials were done in Grange in Meath, and he had a zero in. And actually, I think it was Christina that showed it, that they were over yielding the zero, or at least the same yield as the 350 kilos of nitrogen for the ryegrass. So that is one of the trials. Now we've quite a lot of trials ongoing. We've a grazing platform gone into our dairy and our beef farm. There's some ones going in there, but I think the lowest level is about 80 kilos. But I think as we go forward, we're going to be pushing that as low as we possibly can go. We don't want farmers falling off a cliff in terms of yield either. So we'll go slowly and make sure that we can that the system will still provide what they need.
Yeah, I mean, there's obviously pressures there, because I think we've seen fertilizer use has gone up, I believe, by over 30% since 2012. So that's a huge change. And obviously we've seen that ripple through the environmental system in terms of water pollution and air pollution. And you touched on those in your earlier slides. So I guess that is not, as you described it, the direction of travel. It is the current direction of travel. But ultimately, that, I assume you'd agree with me, that has got to change. And that hopefully that's something, that change will be led and assisted by Teagasc.
Yeah, I mean, I personally would love to see nitrogen inputs go as low as we possibly can make them. And certainly there's some farmers demonstrating that they can go very, very low with them. But we'll do the research and make sure that we can stand over everything before we recommend them more widely. But certainly we can already see we can have substantial cuts to nitrogen inputs and still maintain yields.
Sure. Conor O'Leary asks a question. He says, Fiona, has there been any research done on the trafficability and persistency under grazing conditions of the different sward mixes?
Oh, very good question. I'm not aware of it, but I'd say maybe it's something we can look at on some of our grazing trials. Most of the trials we had done so far, at least in Johnstown, had been cut trials, simulation grazing instead of actual grazing. And this is a research gap. We do need data on graze systems. But UCD have done a trial on sheep grazing in lines. I don't know if they looked at compaction. They probably have some data on that. So might be worth us touching base with them and see if they have something to share on that question.
Okay, well, you heard it first at BioFarm 2020, as they say. Edward has a question for you, Fiona. He says, in the last 3 years, Ireland, we've had 2 significant drought periods. And he would like to know, did the research notice any difference between monoculture swards, such as ryegrass, and the mixed swards? Now, you kind of touched on this earlier, so I wouldn't mind if you could expand a little bit, please.
Yeah, absolutely. And it's a bit of a joke in Johnstown that every time we run a drought trial, we end up with a real drought. And that happened to us actually. So yeah, as I mentioned before, like ryegrass really, really didn't do well in the drought. And I suppose it underpins the vulnerability of the system where you have a monoculture, you have all your eggs in the one basket. And if you get any sort of environmental impact, you could end up with absolutely nothing. So for the same reason that diversifying soil microorganisms or soil biology is really important, diversifying your sward is an insurance policy, because if you end up with some conditions that doesn't favor the one species you have, you're really going to struggle. And to see farmers not being able to feed animals, it's hugely stressful. So I certainly would recommend that insurance policy. Diversify your systems, and they're more sustainable, and they're more likely to stand up when an impact comes.
Sure. I suppose it's always a battle between different philosophies, and one philosophy is maximising the output, and the other philosophy is having a culture or policy of resilience. And it seems at the moment that the output people have got the upper hand, but maybe what you're suggesting that maybe that pendulum would swing in the other direction.
Well, I think we are facing more extreme weather events. We've had more droughts. We're having more extreme rainfall events. I think when we think about systems, we're going to have to think longer than the next productive system, longer than next year, longer than 2 years, 3 years. We need to think about 10 years. And if you have 2 droughts or 3 droughts in 10 years, when you look at the longer term effect, it does make sense to have that insurance policy there. So when the times are bad, at least you have something you still can maintain your production.
Yeah, and I'm not sure that message has percolated very, very deeply, say through the farming press, and that we are dealing with rapid climatic change, and that the certainties that maybe many people grew up with, that they're no longer there. We're essentially everything is in flux, and most of those changes are not in our favour. So perhaps that's something that Teagasc might be maybe a little more upfront about. I appreciate we're only dealing on the scientific side here, but in really articulating the need for maybe greater caution as we lunge forward in expanding certain areas, particularly where that increases our vulnerability.
Yeah, exactly. That's a good point, and I think it's something we could discuss a bit more. So I'll pass that message back.
Absolutely, and I'm not trying to put you on the spot at all. I'll go back to the questions and keep myself out of mischief here. David W. asked the question. He said, what effect does acid phosphate have on limiting microbial access to P reserves? And he wonders, is there any negative effect of muriate of potash on soil biology?
I am not aware of any effect. In terms of the phosphorus question, again, it's something I'd probably want to think about. Maybe I could get back to
Sure.
David with an answer on it. But microorganisms, I suppose, have a phenomenal capacity to scavenge nutrients when nutrients are limited. We do know that phosphorus, and we have some trials ongoing at the moment, phosphorus has a huge impact on the microbial communities present in soil. We've been showing that, particularly if you think of arbuscular mycorrhizae or whatever, they don't do well at high phosphorus levels. But if you're an intensive farmer and you're applying nitrogen to your soil, your nitrous oxide emissions go through the roof if you're at low phosphorus levels. So you have an imbalanced fertility. So the microbial community is very much affected by what you put on in terms of fertilizer, but making sure you have balanced fertilizer if you are applying nitrogen is important. But I'm not sure that I fully answered the question, but maybe I can get back to David another time on that.
Absolutely. Now, I did promise to keep us out of trouble, but I spoke too soon. Siobhan has beaten me to it. She asked the question, protected urea is being promoted as a climate change solution by dairy advisors in Ireland. Is there any research carried out on its impacts on microbial communities at recommended spreading levels?
Well, that trial I just showed you in the presentation I think it's one of the first. It's the first one I've seen in Ireland anyway, that's looked at protected urea. The story with the urea, the protected urea is that if you are losing a large amount of nitrogen in gaseous forms from your system, you're obviously causing quite a lot of damage in terms of greenhouse gases. But we have found that if you switch from CAN to urea, you can reduce your nitrous oxide emissions by 70%. That does have a slight disadvantage in you increase your ammonia emissions. But if you add if you protect the urea, the ammonia emissions go up, but just a small bit. So it's a case of a balance between the two. Overall, the results that we've shown, and as I said, this is preliminary, we will be interrogating the data to the last to make sure that we can stand over everything we were saying. But so far, the indications are the microbial communities don't seem to be much affected. There is some effect on the nitrogen dynamics, but again, that's exactly what the inhibitors do. So I think it's one tool in the toolbox. If you were able to substantially reduce your nitrogen going into your system, I would definitely go for that first. If you're still adding a substantial amount of nitrogen, think about the form you're adding in it and try and reduce down those nitrous oxide emissions, because they are, in the end of the day, related to those extreme weather events that your farmer is going to face next year or the year after.
Yeah, okay, good point. And finally, our last question, Fiona, this evening is from James B. And he says, in your presentation, you suggest different plant communities have different microbial links. Does this back up Christine Jones' theory that a mix of plants will lead to a larger diversity of microbial activity in the soil? And obviously what flows from that is that what we should be pushing, away from monoculture and towards diversity? Absolutely. And scientific research across internationally shows that the more you diversify your above ground community, you diversify your below ground community. And it makes perfect sense because you're creating more niches. I spoke about every organism occupies a specific niche. The more niches you can have, the more organisms you can have, and the more function you have. So what's been shown is the more diverse your communities, the more functions you get from them. So if you have a monoculture, you have roots going down to a single depth, you have the same exudates going out. If you have a mixed species sward or a mixed communities of crops, you have different rooting depths, you have different microbial communities associated with them, you have access to different nutrients, so you end up with a more diverse community. And I think we haven't the full data there, but that seems to be where we're heading with the mixed species swards results, that diversity is there. That's good to hear. Would you, put you on the spot briefly, would you know offhand roughly what percentage, say, of our production would currently be monocultural sward versus mixed? Is it dominantly monocultural sward? Yeah, I would say, well, when I say monoculture, the fact of it is people may plant monocultures, but I suppose over time, if they're long-term pastures, there probably is more species going into them. I think now you have to plant, if you're planting ryegrass, you have to plant a low amount of clover with it. But whether that clover is maintained in the sward depends how you manage your sward. Obviously, if you're putting high levels of nitrogen, your clover will disappear out fairly quickly. My understanding is a lot of the reseeds would be mainly monocultures or monocultures with a bit of clover. But I'm really thrilled to see around the country there's a lot of farmers who are trying out mixed species swards. Like ourselves, they're probably feeling their way, trying to see what works, growing an acre or two and seeing if it works. But I think this is really positive. It's still an intensive system, but it brings in a bit more diversity. Yeah, and I guess diversity in the end of the day is the name of the game, because this is farming with nature. Exactly. Okay, so I think that concludes this session, Fiona. So just to say thank you very much for your time and for your excellent presentation. Pleasure. Thanks very much. All the best.